Hello, my name is Dr. David Parrott. I'm a biology professor at Westminster College and the assistant director of the Great Salt Lake Institute. Today I'd like to talk about my research into the shoreline plants that grow out at Great Salt Lake and their unique rhizosphere microbiomes. So my research really has three components to it. First of all, the plants that grow along the shoreline. And these were really chosen because these live in a really extreme environment. If you consider the lack of water, temperature, and soil salinity. Salt is a big component here too, because we have this terminal lake that we live by. It's shrinking because of water diversion and our ongoing drought. And so soil salinity and these plants were really intriguing to me. How do these plants really survive in the salty conditions? But as a microbiologist as well as a plant biologist, I'm really interested in the interaction between the microbes and the plants in these salty soils, because these microbes may contribute to a more favorable environment for the plants to thrive. And in a drying West or a drying world, this kind of information could be really important to us in the future. So to get going, let's start by talking about the lake. It's been in the news quite a bit. I'm sure most everyone has been out to it at some point or knows of it. But the lake is a really unique environment. And what makes it really unique and interesting from a research perspective is that we've got these two parts to it, these two arms, the north arm and the south arm. They're separated by this roadway, or railroad causeway rather, and the railroad causeway really bisects the lake. Freshwater is coming in to the south arm, and so this reduces the salinity of this terminal lake, but it's still quite saline. And then the north arm is isolated for the most part and becomes hypersaline. In fact, typically the salinity of in the south arm is between 9 and 15%, although right now it's higher than that, which is stressing those organisms that live in the lake. And then the north arm is really at saturation, so between 24 and 30% salinity. What this means is that shoreline plants are exposed to these hypersaline conditions, either in the south arm or the north arm, and as the lake shrinks, it leaves behind some of the salt, which is incorporated into the soil, and this creates two different soil salinities, north arm soil salinity and south arm soil salinity, that plants then have to adapt and thrive in. So let's go ahead and talk about plants. The plants at Great Salt Lake are not unique in the world or rare, but they're really very interesting, typically small, and can tolerate really quite astonishingly high concentrations of salt in the soil. So looking back, there have been many researchers over the decades that have been looking at vegetation in and around Great Salt Lake or the Great Basin. And a lot of really interesting contribution came from Seville Flowers in 1934, when he did quite an extensive survey, at least in my in my realm of shoreline plants, and really sort of talked about all of these areas. But there have been many contributors to the research of those plants that grow around the lake. So we can look at different kinds of vegetation and just in and amongst the lake, and really quite diverse. But what really sparked my interest is those plants that can tolerate the highest salt concentration in the soil. These were the most extreme and really gave sort of the most extreme example of adaptation to really extreme environments. And so we can look at a cross-section of the shoreline, and really my area of focus is for plants and for microbes is down in this pickleweed zone over to the left-hand side. As the lake is shrinking, we're getting a lot more playa. lake bed being exposed. And this is so salty that it prevents almost anything from growing in it, and especially plants. And so I look right at the very edge, that interface between where pickleweed can grow and where it can't. And this is really my area. Now, shoreline is really kind of a weird thing because the original shoreline is far, far away from the water now as the lake shrinks. But because the playa is so salty, nothing will grow there. So I call them shoreline plants, but they're really, far away from the shore. So really we look at two different genera primarily. The first we look at is Salicornia. So pickleweed is fairly common in and around the edge of the lake, depending on how salty the soil is there. And so I really search out the saltiest of salty soils. And so we are really looking primarily at this, but we also look at other plants like iodine bush, for instance. This doesn't grow in the same area that Salicornia grows, although Salicornia can tolerate less salty soils, but we're really looking at the very edge, again, of high salinity. We also find that seepweed is found in amongst some of the pickleweed. And so again, we're looking at those seepweed that can really tolerate the highest salt concentration. And then finally, we look at a lot of different grasses that grow in and around the shoreline. These have a tendency to be a bit upslope or away from the pickleweed zone, but are still of interest to us. So let's go ahead and talk about the shore for a second. Really, when we talk about the shoreline, we're talking about the ancestral shoreline, where the water would have been when the lake was, fully filled, I guess. And of course, this varies constantly. But the shoreline is fairly distinct in that, depending on where you are, like in the North Arm, you can see where plants will grow and then where plants won't grow. And there's a fairly sharp distinction here because of lack of water availability and the hypersaline soils. There's really a distinct area these plants will grow. This is a little less noticeable in the South Arm, so around Antelope Island, where we get a bit more plant diversity down at the shoreline. So we see a bigger abundance of different genera and species than in the North Arm along the shoreline. And what this really equates to in my research is a difference in the makeup of the microbiome around these plants at these two different locations. So let's talk for a minute about the microbes, and let me just give you an overview of what our collection, what our research is really all about. So we have two different locations that we sample from, one in the North Arm and one in the South Arm. We identify the plants that grow right at the very edge of the super salty environment. So what would be the traditional shoreline, where we look at the soils, we test salinity, we dig around the roots and isolate microorganisms. We do this in a traditional microbiology way where we actually look at the microbes growing in traditional microbiological media, but we also extract DNA from the soil and do a much deeper analysis by looking at what microbes are there, even if we don't know how to grow them in the lab. So again, we have these two different locations where we sample, and we've been doing this since 2018. We do this on a sort of seasonal basis, so we go out in the early summer every year. We sample around these plants. We sample a number of them at a few different locations at Sparrow Jetty and a few different locations at Antelope Island, and then we spend the rest of the summer in the lab extracting DNA from the soil and growing microorganisms. Those microbes look a lot like this. They look like soil microbes. They're pretty smelly. These are growing on a media that favor those microorganisms that like growing in salt, so that's what the 12% is. So these are the microbes that are found typically at Antelope Island. these at the spiral jetty. And we see that there are different kinds of organisms there. And there's some overlap, but they have very distinctly different microbiomes between the two pretty salty south arm and the super salty north arm. If you look at just the bacteria between Antelope Island, which is the AI there, bacteria AI versus bacteria SJ, which is spiral jetty, there's a bigger assortment of orders of bacteria that grow in the lower salt concentrations in the soil around and on Antelope Island versus those at Spiral Jetty. There's less less diversity, and this changes quite a bit when you look at the archaea. Archaea are a extreme version, if you will, of bacteria in a sense. And these inhabit hot pools and extreme chemical environments. And in the extreme salty environment of Antelope Island, we see, some pretty big diversity, but we see hardly any diversity of archaea at Spiral Jetty. There are just a few main orders of bacteria that grow there. So we've expanded our research a little bit to incorporate fungi as well, and so we can look at bacteria, archaea, and fungi, and we can see differences based on location around the roots of plants in Antelope Island versus Spiral Jetty, and we can see differences, and we can look at these sort of dramatic differences in certain cases where some orders of fungi are overrepresented, say in Antelope Island, where they're only a small percentage of the overall at the Spiral Jetty. And so we see this interesting makeup at each one of these locations. But what really is fascinating about what we've found more recently is that around the roots of particular plants at each one of these locations, we see a specialized microbiome. So the microbiome around pickleweed is different than the microbiome around seepweed. And it's different, too, from the microbiome of bare soil that has no plants growing in it. And so we can look at this in bacteria and in the archaea, and again in fungi as well. And as we begin to pick apart who these bacteria are, we're starting to see that there are certain orders of bacteria which traditionally have been found to aid in drought resistance or drought tolerance in some plants and in metabolizing heavy metals and salts and things like this. And so our research is ongoing, but it's really exciting and encouraging to find these particular orders of bacteria, fungi, and archaea that could be beneficial to helping plants survive in these super salty soils. So I'd like to wrap up by thanking my students. At Westminster, we are an undergraduate primarily institution, and all our biology folk are undergraduate researchers. We've had a bunch of them run through the lab and contribute to this project, and it's been a lot of fun and very interesting. And it continues on to today. We're going to be doing a little bit more work on salt this summer and looking at more of these microbiomes. And finally, I'd just like to thank Westminster College and all of those contributors to help fund my research. And I would certainly be happy to take any questions that you have. So thank you very much for your attention.